This video is part of a course on food physics, of which this is the theory section. This module is about engineering descriptions of food safety. Now, it is important to note that food safety is a discipline by itself, and there are many ways to go about discussing it quantitatively. This video is simply one quantitative approach, one quantitative framework that should resonate with an engineering mindset. The modules before this discussed various frameworks for process physics and food quality. And this module concludes the engineering framework for food physics for most conventional heating processes. This is part one of a four-part video series discussing the safety framework. But this video is simply providing an introduction or the big picture. We are going to talk about two different types of food safety framework. First one, we'll talk about microbial safety framework. For example, how do I get a good quantitative picture of the bacteria that might be there inside the meat, for example, during the cooking process? Does it survive the cooking process? Then the other type of safety that we'll talk about is chemical safety. For example, in this frying process, as the potatoes are heated at high temperatures, there are carcinogens that form called acrylamide, and we want to know how much the forms, at what temperature, at what time, and so on. So that would be called chemical safety, and the other one I discussed would be called microbial safety. And in the subsequent videos in this series, we're going to get into details of them. Here we simply introduce the overall framework for these. In the quantitative treatment of microbial or chemical safety, or an approach to microbial or chemical safety prediction in a mechanistic way, the main idea is to treat the microbes or the chemical components inside the food as a chemical species. By treating them as species, it readily falls into the engineering framework already developed for species mass transport. That's the mass transport equation. To recap, you may recall from earlier videos or from your mass transfer course, you did a control volume where you did a mass balance for a mass species. And so this control volume, which is the red boundary here, you say that what's coming in and what's going out of the control volume, if a mass species is generated, leads to change in storage of the species. And so they are related this way: in minus out plus generation equal to change in storage. So this is always true. This is simply conservation of mass for that one species. If you follow through with the derivation, you get an equation that looks like this, where all of the terms have physical meaning. So this one is kind of a net change in storage or rate of change in storage. And then this one is the net change due to species convected in, and this one is net change due to species diffused in, and this is the net change due to the generation. So you did that before and you are familiar with the transport equation. So what is that doing here in the context of a bacteria? Diffusion, for example, can be due to random motion uh, or motility simply because the bacteria is a living thing. Diffusion can also be due to Brownian motion from just having energy. So it's basically random motion. That's what this term stands for. The convection or flow term can be simply due to passive motion where the bacteria is carried by the fluid in the food, or it can be due to things like when the bacteria is searching for a food, or chemotactic movement. The generation term for the bacteria can simply be the growth of the bacteria, or it can represent, it can be a negative generation term. For example, it can be due to inactivation of the bacteria. And these generation or growth of the bacteria, inactivation, they're the subject of microbial kinetics. That was about bacteria. And just like bacteria, then for chemical components, the carcinogens can also diffuse, or they can be carried by the fluid, and more of them can be generated. So the same transport equation can help us compute in a quantitative way, in a mechanistic way, the microbial changes or carcinogen related changes. So let's call this microbial and carcinogen physics that can provide concentration of chemicals or microbes or chemicals as our safety prediction framework, or microbial or carcinogen physics. The microbial changes that we just talked about is not going to happen at the same rate everywhere inside the food. And let's find an example, a very simple example to make the point. So let's take canning of these peas in brine. So it's a thin fluid that when I heat the can from both sides, then the fluid is going to be heated and it's going to change its density. That's going to set up flow pattern, and the fluid is going to move like this. So if it does, then we know that the temperatures are going to be higher here because it's closer to the wall, and lower here because it's farther away from the wall, plus complications due to flow. So most obviously the bacterial destruction is going to be more here and not as much here deep inside. And that makes the point that the bacterial destruction is not going to be same everywhere. So to find out how much is the bacterial destruction, we basically have to combine the bacterial kinetics with what's going on in terms of the process. Likewise for carcinogen, again we have to combine the two physics. So we have to combine for each scenario either the microbial or the carcinogen physics with the process physics to get safety prediction. So again, to repeat, the microbial or the carcinogen physics will need to be combined with process physics to get safety prediction. That is the main idea for a framework, a quantitative, a mechanistic framework for safety prediction. The food safety framework that we just discussed, if we want to put it in perspective, and if we look at the big picture of food safety, we can think of it at four different interconnected complementary scales. We can do subcellular level modeling or subcellular scale modeling to reveal the underlying mechanisms of biochemical processes that happen inside a bacterial cell. That is not what we will talk about in the subsequent videos, but just to show that that is another scale that is possible. The, the next higher scale is the microbial population scale. Here we can talk about modeling of environmental factors like temperatures on bacterial cell and a population in a culture medium. This could be, for example, a growth model. This is also not what we will talk about. This is very well developed field also, and we will use the empirical or other growth information that is already available and included in our framework. The next higher scale, we call it the food scale. At the food scale, we combine the descriptions at the lower scale, like the growth, for example, with descriptions at higher scale. And this is what this multi-part video is about. Finally, this is the largest scale. At this still higher scale, we can use the information from food scale. So, for example, we can use the information from the food scale and combine with how much am I eating? And then what is the probability of me getting sick for a certain level of bacterial intake? So then this is for one person, then we can scale it up to the entire population and find the probability of sickness for the entire population. So loosely speaking, what I just described at this higher scale is called risk analysis, and it can be thought of food safety as the highest scale. And so for this we have actually a separate module if you're interested, but we will not be talking about it here as part of this four-part series of framework for microbial and chemical safety. To repeat then, we will be talking food safety at this food scale, and we will not be talking about these lower scales or that even higher scale at risk as part of this four-part series.